ITACA was an investigator-initiated trial based on the pharmacogenomic-driven concept. Why ITACA and why we decided a long time ago to design this trial in the adjuvant setting? Well, because the advantage from the platinum-based chemotherapy in the adjuvant setting is very modest. So in our experience, we used ERCC1 and thymidilate synthase starting from a pilot trial as a biomarker to design a clinical trial in the adjuvant setting, completely resected the stage 2 and 3 patient with non-small cell lung cancer were randomized to receive a pharmacogenomic-driven treatment versus the standard of care. The trial was conducted in 31 centers, mainly in Italy and Germany, and 1 center in Poland. And the primary endpoint of the trial was the overall survival. So unfortunately, we were not able to reach the primary endpoint. Even if there was an advantage in terms of survival of the pharmacogenomic-driven arms compared to the control arms, this was not statistically significant with an hazard ratio of 0.76. And at the same time, also among the secondary endpoint, this is, there was an advantage for the time to relapse in favor of the pharmacogenomic-driven, but also this result was not statistically significant. At the same time, there was an interesting result concerning safety, because we registered an advantage in terms of toxicity profile for the pharmacogenomic-driven arms compared to the control arms, mainly focusing on the hematological toxicity. And if you want, this is an important result in the pharmacogenomic context. So unfortunately, the results were not statistically significant and the primary endpoint was not met. But at the same time, there was this advantage in terms of overall survival and recurrence-free survival. And this was already presented during the World Lung Cancer meeting in 2015 and confirmed this year, these results concerning safety. So I strongly believe that these results will be relevant in designing future trial and the future therapeutical approach in the adjuvant setting, even when we will be able to implement the use of immunotherapy or immunotherapy plus chemotherapy in this setting.